Hello, in this video I'll be talking about how cone snail toxins affect cell signaling. First of all, we need to know what a cone snail is. Cone snails are marine mollusks that are typically found in warm tropical waters. They are carnivorous and hunt and eat worms and fish. Now, one might think that in order for a snail to be carnivorous, it would consume dead organisms or hunt other snails and mollusks, because snails are notoriously known for being slow. But cone snails, despite being no exception to being slow, are unlike most other meat-eating snails in that they capture live prey, and they do this with the help of venom. Their method of delivery for this venom is also very interesting, as long before humans started using hypodermic needles, cone snails have already evolved specialized teeth called radular teeth, and they are essentially tiny harpoons that can be filled with venom. These harpoons are stored in a specialized organ in the snail, and are filled with venom when the snail brings it into its proboscis in preparation for a hunt. The proboscis is like a dart gun, firing the venom-filled harpoon via muscle contractions. And once the harpoon has been inserted into a prey item, say a fish, the fish will become paralyzed almost instantly, and the snail pulls the fish into its mouth for consumption. Cone snail venom is made up of a cocktail of multiple different peptides, up to 100 different kinds, and can vary from species to species and even from snails of the same species. Despite this, one thing most peptides in the venom have in common is that they interfere with ligand-gated ion channels, most prominently in the neuromuscular junctions. Normally, motor neurons deliver signals to skeletal muscles through neurotransmitters. The axon terminals of motor neurons are connected to skeletal muscle fibers, and they secrete neurotransmitters, acetylcholine, to the muscle through a synapse. First, calcium channels on the motor nerve end are opened by the motor neuron's action potential and absorb calcium ions. This causes the acetylcholine-filled vesicles in the neuron to fuse with the neuron's plasma membrane, releasing the acetylcholine to the synaptic cleft, which is the space between the axon terminal and the target cell. From there, the acetylcholine binds to the acetylcholine receptor proteins, the ligand-gated ion channels, and opens the protein channels, allowing sodium ions to flow into the skeletal muscle. So the acetylcholine is the ligand, and the acetylcholine receptors in the skeletal muscle fibers are the receptors. Once the sodium ions enter the skeletal muscle, more sodium channels in the muscle fibers are opened, depolarizing the fiber, producing and spreading an action potential. The action potential triggers a chain of enzymatic reactions that open calcium storages in the skeletal muscle's sarcoplasmic reticulum. The presence of calcium changes the shape of actin filaments, allowing the myosin to bind to them, causing a contraction. So here is the cellular end response, a muscle contraction. Almost as soon as the acetylcholine binds to the receptors, they are removed by an enzyme called acetylcholine esterase. The sodium channels close back up, and the potassium channels open to restore polarization and end the action potential in the muscle fiber. The absence of the action potential stops the release of calcium into the fiber, and actin reverts back to its original shape, relaxing the muscle fiber. That is how muscle cells normally react to the signal from the brain, but in the presence of conotoxins, many, if not all, of the ion channels are blocked by the multitude of peptides. Because cone snails can't afford to have their prey resisting their toxins, as they wouldn't be fast enough to recapture the prey, they need the toxins to immediately immobilize the prey. While a toxin will usually take a couple of seconds to take effect, cone snail toxins can immediately paralyze a prey item with such a rich variety of peptides present in the venom, targeting different parts of their nervous and muscular systems. One of the peptides present in the venom is omega-conotoxin, also known as Prialt. This peptide blocks the calcium channels in the axon terminal, preventing calcium from getting into the nerve ending, which prevents the release of acetylcholine. As a precaution, there are many other peptides that block the other channels involved. For example, alpha-conotoxins bind to the acetylcholine receptors on the skeletal muscle, so that even if acetylcholine is released, the alpha-conotoxin prevents the acetylcholine from binding to the receptor. Another conotoxin that often accompanies alpha-conotoxin is psi-conotoxin, which plugs the receptors, doubling down to make sure nothing gets through them. If somehow these three types of conotoxin aren't effective and all the ion channels work as intended, another type of conotoxin called mu conotoxin comes in and plugs the sodium channels around the muscle fiber, so that even if the action potential is able to pass from the motor nerve to the muscle fiber, it can't spread. With the action potential being unable to spread through the fiber, the muscle won't know to contract, thus resulting in paralysis in the prey item. So that's how cone snails have evolved to become slow but successful predators, by preventing the brain signals, and thus the ligands, from reaching the target muscle cell, blocking receptors and preventing the signal transduction from starting.